Good morning. I'm a researcher working at the Medical Genetic Unit of Neurovascular Institute for Rare Disease. And since several years, I'm interested in the Desmoid tumor research. And our project has been supported by the Desmoid Tumor Research Foundation. It's my pleasure to be a part of this workshop, and I will present our study regarding the CRISPR-Cas9 system to create a desmoid-like cell model to study their response with target stimuli and the cell-cell interaction. Our studies on primary cell isolate from different bioptical samples show that different desmoid cell respond to standard microenvironment stimuli with a high degree of variability. In particular, EGF-beta1 play a role in cell proliferation, cell spread, and myofibril penetration of desmoid cells, suggesting that the cellular sensitivity to EGF-beta1 is an intrinsic feature of desmoid cells. Moreover, that conflict is a critical determinant of EGF-beta1 induced in the desmoid myofibril differentiation, and it regulates cell-cell and cell adhesion interaction in desmoid myofibril differentiation, suggesting that the intercellular communication have an important role on the desmoid myofibril behavior. Our aim is to study the effect EGF-beta1 stimulus in cellular communication, and for this we are looking to the cell-cell interaction, the cell matrix adhesion, the role of cell adhesion receptor, the focal adhesion formation, and the expression of integrin receptor. It is our aim to create simple desmoid-like cell model that ensure genomic and phenotypic stability. For this purpose, we use the CRISPR-Cas9 system to induce efficient, specific, and stable gene editing in the Wnt gene to generate the cell model that ensure that will mimic desmoid cell behavior. We found that integrin are expressed in desmoid cell and are evolving in crosstalk with EGF-beta1. EGF-beta1 indeed EGF-beta1 controls the transcription of genes that encode several integrin, and it affects the cell adhesion and migration by regulating the expression of integrin, their ligands, and integrin-associated protein. Furthermore, integrin can indirectly control the expression of components of EGF-beta1 pathway. The crosstalk between integrin and EGF-beta1 is crucial in fibrosis process. Immunofluorescence data of normal and desmoid cell untreated and treated with EGF-beta1 express different integrin. In particular, alpha-11 is expressed only in desmoid cell, while alpha-V and alpha-5 integrin are more expressed in desmoid cell after EGF-beta treatment. On the other hand, alpha-2, beta-1, and beta-3 integrin are expressed in normal and desmoid cell independently from the treatment. Integrins link the extracellular matrix with the cytoskeleton, forming a complex network and focal adhesion formation assemblies. The focal adhesion assembly are a peculiar structure in myofibril differentiation, and EGF-beta treatment promote the focal adhesion rearrangement. So, what is the role of EGF-beta in desmoid cell focal adhesion formation? Immunofluorescence data show that the expression of post-adhesion kinase show a strong staining in focal adhesion site of EGF-beta of desmoid cell, more expressed after EGF-beta treatment. Immunofluorescence data of desmoid cell after EGF-beta treatment show a fibrillar structure localized with the focal adhesion sites, more evident after EGF-beta treatment. To better analyze the behavior of the desmoid cell, we plan to generate a uncontrolled cell model that will mimic the desmoid cell by using the CRISPR-Cas9 system. We plan to create a knockout of APC gene and a knock-in of beta-adenine gene. And for this purpose, we use the plasmid Cas9 to aid promycin. We performed preliminary experiments to optimize the CRISPR-Cas9 protocol. First of all, we tested the 3 cell line HSF1, Detroit, and VJs in fibroblasts. And looking at the cell growth and the cellular culture condition, we chose the VJ cell line for the prospection experiment. Afterward, we set up the optimal promycin concentration for the cell selection, and the data of cell survival by MESA show that the best treatment is 42 hours with 2.5 micrograms per ml of promycin. For the prospection protocol, we tested the electroporation and the lipofectamine protocol. And by using 10 microgram ml of plasmid and 105 mol of gig, we choose the lipofectamine as a prospection protocol. As after electroporation, most of the cells were divided. The respective and selected cells were then isolated by several serial dilution or limiting dilution. For the APC knockout gene, we selected 4 different DNA genes localized in the exon 4, exon 9, and exon 16. Optical morphological analysis show that APC knockout VJ cells show heterogeneous cell phenotype comparing to the wild type, with seeing as small cell center. The APC knockout was also evaluated by sequencing analysis of the transfected gene and immunofluorescence analysis showing the absence of APC signal in APC knockout cell. Selected the APC knockout VJ cell treated with EGFR show a fibrillar cytoplasmic structure localized with the focal contact size of the cell, as observed in primary desmoid cell, suggesting that EGFR play a role in APC knockout VJ cell proliferation and myofibroblast phenotype. For the knock-in of beta-adenine gene, we selected 2 CRISPR genes of DNA localized specifically to the phosphorylation site, and in particular the threonine 41 and serine 45. And we selected several donor template sense and antisense. The beta-adenine in VJ cell a week after promycin selection restarted to grow, showing first an increased cell size, afterward increased cell number, but with a low proliferation rate. The optical microscope analysis show a heterogeneous phenotype comparing to the wild type VJ cell with smaller and thin cell feature. Immunofluorescence analysis of the beta-adenine knock-in in VJ cell show a strong and specific nuclear localization of beta-adenine, differently from the wild-type cell where beta-adenine is cytoplasmic. In conclusion, the APC knockout cell respond to EGFR stimulus like desmoid cell. The beta-adenine in cells show the desmoid like cell phenotype showing a strong nuclear signal as a desmoid primary cell. EGF-beta1 promotes propagation localized with similar structure in desmoid cell. In the future, we will investigate the role of EGF-beta in a single-out and beta-adenine cell. We will study the cell-cell interaction in the apical out and the beta-adenine cell. We will investigate the crosstalk between EGF-beta1 and focal adhesion in this pathway, and moreover, we will study the role of the wild-type cell in the role of the apical out and the beta-adenine cell. Finally, I want to give a special thanks to the founders Anna and Giuseppe Baschinotto, to the melanoma unit for the biotic sample and clinical support, and to all my colleagues, in particular Anna and Marco, that made most of this work.